Hey team, Dr. Jake Order here, and this video is a really good one because I'm going to take you through drug development, and I think it's so important for everyone trusting the systems of science to understand the drug development process. I'm going to use antihistamines as one of my examples just because I'm covering the immune system at the moment, but I'm also going to look at the COVID-19 vaccine by AstraZeneca as another little example. I'll discuss that at the end of the video. Here are some antihistamine drugs here, commonly taken all over the world to block our inflammatory allergic response to things like pollen during summer. So let's jump into it. Now, I'm going to jump into modern drug development. Back in the day, drug development was a bit different. Here's Edward Jenner. He came up with an idea on how vaccines could work. He sort of discovered the modern vaccine, although it's a bit debatable. And he just came up with the idea and decided to run it on the 8-year-old son of his gardener. So they just jumped straight into humans, often children. Louis Pasteur did this as well. He did this with children. And then after a couple of children died, Louis Pasteur said, I better go test that on dogs first. So it's what I'm going to take you through right now. The point of this is modern drug development, not historical drug development. So this is what modern drug development looks like. It starts with cell cultures in a dish most often. So we have cell cultures in a dish, and in that cell might represent a cell type that we're interested in. If we're interested in blocking the histamine response, we would put cells that are sensitive to the histamine response. So maybe blood vessel cells, because blood vessel cells respond to histamine. We could put histamine on those cells and see if we can block it with our drug. So the key is to set up a high throughput drug screening process that mimics a biological process that we're interested in. Now, I've got a flask here. There's a flask of cells grown, but often we use plates with 96 wells in it, so we can now test 96 different things simultaneously. In fact, you can even go up to 384, so 384 wells in a tiny plate like this. It requires some very masterful pipetting, although robots are now doing a lot of this pipetting too, to test hundreds of drugs at a time sometimes as well. Now, we take the results from that and we actually plug that into computers, and we're now using this more and more and more, especially seeing as Google's AI has now cracked protein modeling. I just looked that up. That's super cool, and it's going to revolutionize medicine. But anyway, the point is we know the structures of the molecules we're going to target, so now we might be able to design drugs to better target that protein using in silico computer techniques. But often these feed back and we go round and round and round several times through this process. So we'll screen a bunch of drugs, we will find a lead candidate, we will then plug that lead candidate into our computer model and see how we might be able to improve it. So then we would generate a bunch of drugs that are similar to that lead candidate, and we would screen them again in our cell culture. Now, this would actually inform the computer program about how the tweaks worked. So if you change the molecule like this, it improves it. If you change it like this, it makes it worse. The computer can now make more informed decisions about how to make it even better than it already was. So we go round and round in this process until we get a really optimal drug. And this is called structure activity relationship processing. And it's an excellent way to make an excellent drug. And then once we get our lead candidate, after screening it on hundreds and hundreds of cells, we need to go higher, right? Now, what I want you to imagine is I come up to you and I go, I think I've got a new drug to treat your disease. And you're like, great. Can I, and I go, can I inject it into you? And you go, what's it been tested on? And then I say, well, actually, you're going to be the first organism that we give it to. Are you going to take that drug? Are you going to be the first person to be exposed to a drug ever? The first animal? The first anything to be exposed to the drug other than a single cells in a dish? I don't think so, right? It's kind of a scary idea. And so this is why we need animal research. We need to understand how it's going to affect the whole body. Now, cell culture is great, but we cannot get the hundreds upon hundreds upon hundreds of cell types and cell interactions modeled in cell culture. We need a whole organism. You need a liver to know if it's liver toxic. You need a kidney to know if it's kidney toxic, right? So we really do need whole organisms, and it's a sad state of affairs, and I can't wait until we're beyond that, but right now we are definitely not beyond that. So the next thing is we've gone in silico, which means silica is silicon chips. It's the computer chips are made out of silicon, so we call it in silico. In vitro literally means in glass, and that's because it's in glass dishes. And in vivo means in life. Vivo, vivacious, and vive la France and stuff like that. That means life. In vivo means in living animals, right? So these are the three main branches of what's called preclinical research. So this was before it has gone into the clinic. So we take our lead compounds there, and typically we like to test it in at least two very different species. Sometimes we go into species that are more genetically similar to humans, such as ape-like species, before we go into the human condition. But then often with this, we also generate the disease that we're perhaps interested in these models. In a previous video, I talked about how we generated an Alzheimer's mouse model by taking a gene that causes Alzheimer's disease and inserting it into mice, and then those mice then develop memory deficits just like Alzheimer's disease. So often we generate the disease in these animals and then treat it with the drug that we have developed through the in vitro and in silico processing. We then take it into clinical trial. Now, this is quite a journey. This is a massive journey to get through this process. And it literally takes billions of dollars to get from here all the way through to clinical trial to medical treatment. It takes a huge amount of money to get through. Now, clinical trial isn't a single stage, and this is why it's costs a heck ton of money, and it is a multiple stage process. So we have phase one. Phase one is typically done in tens of people, and the idea is to test principally, is it safe? Now this is done in young healthy males, and there's a bit of a saying, I don't know if I should say it, young, dumb, and full of semen, is what I will say. And that is not far from the truth. Why do we test it on males? And it's because this is the explanation often given. Males reproduce sperm regularly, whereas women, when they are born with the predecessor of all their eggs, right? So they have essentially they're born with all the eggs they will have in their life. They don't make new cells that make eggs, okay? And so if you wipe out those cells, if your drug has a surprise interaction and wipes out all those proto-egg cells, the woman will become infertile. If you take a drug that wipes out all the sperm, the man will just create more sperm. Now, so we typically, in a phase one trial, we take healthy young males and we give it to a low number of males, and we just measure, did it cause them to drop dead? Is it acutely toxic? And so we don't need a lot of numbers to see, is it acutely toxic? Now, we then might go test it on a low number of females as well to make sure it is safe in females, and then we would go into a phase 2 trial. Now, phase 2 clinical trial is typically hundreds of people. And in it, we're basically saying, is the drug doing what we expect it to do, right? So if the drug was designed to block histamine, does it block histamine in these people, for example, right? So we do that. Now, typically, we don't have the numbers in the clinical trial. We don't have the statistical power to test, is it truly good? Does it make a clinically meaningful change in the disease or in the patient, right? But we can measure a proxy measure of efficacy, right? So, for example, if it was the histamine thing, we could look at something like vasodilation. Did we block vasodilation with our antihistamine? We know histamine causes vasodilation. Did we block vasodilation? Now, we're not measuring a clinically relevant outcome there. We're just measuring vasodilation. So is the drug acting as expected? Did it block the histamine response? Did it block vasodilation? This is all phase 2 clinical trial. It's typically done in males and females, and it's typically done in the target population. So if your drug is designed to cure hay fever, it will be done in people who have hay fever, right? So if it's an antihistamine, it will be done in hay fever, and its ends are in the hundreds. Then after that, we jump into the phase 3 clinical trial. Now, this is us answering the question, does the drug cause a clinically meaningful improvement? Now, so Clinically meaningful, what does that mean? It could mean reducing heart attacks, it could mean reducing deaths, it could mean reducing time in hospital. For antihistamines, maybe it means reducing time off work. It just means making a meaningful impact. What do you want the drug to do? Does it do it? So instead of using a proxy measure like vasodilation, you might actually use a relevant measure like days with a runny nose for hay fever. Could be a clinically meaningful outcome. Does your antihistamine reduce the number of days the patient has a runny nose? That is a clinically meaningful outcome. It's again done in the target population, both males and females, and it's typically thousands, and this is where the cost can start to go through the roof. Then after that, we have what's called phase 4. Now, not a lot of people talk about phase 4, but it's incredibly important, and we're seeing it in the news right now. Now, phase 4 isn't really a formal paid clinical trial. What it is that governments around the world have set up an infrastructure to monitor side effects of various drugs or monitor adverse events of various drugs, and it allows us to model thousands, if not millions, of patients taking those drugs to see if there are any surprise side effects once we get to a bigger cohort and once we monitor them for longer, right? So a phase 3 clinical trial may not be that long. It may be half a year, maybe up to 5 years I've seen with some clinical trials. But phase 4 could be decades, right? Because really it's just government monitoring of are drugs okay? And it's so important that we have that network set up, as I'm about to discuss. So we mentioned going from preclinical to clinical. I think it's so important people understand this, and I don't think it's genuinely understood very well. But the translation of preclinical research to clinical efficacy is terrible. So let's have a look at cancer, right? So for every 1,000 drugs that have passed cell culture, passed in silico, and have gone on to animal trials and have passed animal trials. So for every 1,000 anti-cancer drugs that work in animal trials, around about 300 will get tested clinically. And of that, only a few will get through phase 1 clinical trials. Do they really meet that threshold of being perfectly safe in a human? Then a very small number will get through phase 2, very small number will get through phase 3, and a very small number gets through FDA approval. You can't even see it on the graph. That's how dismal the translational success of these drugs are. So I'm going to put it to a log scale. So this is what we call a log scale. It allows you to look at numbers for 1,000 and 101, which is nice. So we can see for every 1,000 drugs that work at treating cancer in animal models, 300 will get tested clinically, 25 will get through phase 1, 10 will get through phase 2, 5 will get through phase 3, and only 2 will end up with FDA approval and taken into the clinic, so 1 in 500. And that is dismal, isn't it? When we think about it in terms of getting all the way through that research pipeline. This is another reason why it costs billions upon billions of dollars to end up with an effective drug in the clinic. And it's one of the reasons why drugs cost so much, is it costs billions of dollars to get them through the clinic, that when you get them through, you sort of have to make your money back on all those failures in your research. Now, this is called a translational failure, and the preclinical researchers often blame the clinicians, and the clinicians often blame the preclinical researchers. The preclinical researchers might say something like this: we tested it in these conditions that were very narrow, very specific. An example might be for stroke. Say you're having a stroke, which is a clot in your brain. We tested it if the drug was given immediately, 15 minutes after the stroke, and it was only a very specific kind of stroke in healthy males, for example. That was the preclinical trial. You then took that into the clinic and you gave the drug 6 hours, 10 hours after the stroke. You did it in morbidly obese people. You did it in females and males, and you did it with hemorrhagic stroke, which is a bleed in the brain, and ischemia, which is a blockage in the brain. And so the preclinical research is saying, we never said it was going to work under those conditions, but you ran the trial anyway and it failed. Of course it was going to fail. The clinical researchers rightly point at the preclinical researchers, and what they should say, they shouldn't say animals aren't humans. That's not what they should say, because if you look at a microscopic slide of our liver, you're not going to know if that liver came from a mouse or a human. We are all kind of like Lego. We're built from the same parts. We look pretty different on the outside, but on the inside, we are very similar. I would say that the difference between mice and humans is massively overstated. If you take any drug that works on a human and give it to a mouse, it works in the mouse. If you give a mouse caffeine, they become alert. If you give them Ritalin, they become hyperactive. For example, if you give them, cocaine, they can get addicted to cocaine. Like any anti-cancer drug that works in humans will work in the mouse model. So what's going on there? Probably the answer is poor quality preclinical research. Not blinding, not randomizing. All these things to do with quality of the research is really poor in the preclinical. And we need to take a long, hard look at ourselves, and we are, about improving the quality of our research. We should be aiming for the same standard as clinical research, which is blinded, double-blinded, and multi-centered, which means it's not just happening in one lab throughout the world, it's happening in labs all over the world with really high N numbers. Much more powerful research needs to be done, but there are failures going in both directions. Anyway, so now let's talk on to the kinds of drugs you can develop. Now, there's two main groups of drugs that we like to talk about. There are biological drugs, often called biologics, and there are small molecule drugs. Now, small molecule drugs are what we commonly think of. Paracetamol, aspirin, ibuprofen, all of these are small molecule drugs. And they're the vast majority of drugs, to be honest. But then there are biologic drugs. Now, biologic drugs are made out of biological molecules, often proteins. And a good example of that is a bunch of antibody drugs. So we talked about antibodies and how antibodies can bind to certain things. Well, what if I wanted to block, say, a cytokine, say interleukin 1? Interleukin 1 is a protein cytokine in my body and it signals inflammation. Well, if I take human interleukin 1 and I inject that into an animal, that animal will actually have an immune response to it because it's not a self-antigen. So it will treat my IL-1, human IL-1, as a pathogen, and it will generate antibodies that will bind to that IL-1. It's a bit like vaccinating an animal for human IL-1. They will produce antibodies for human IL-1. We can then take those antibodies. Now we have a protein molecule that will bind to our IL-1. Now, if you bind to IL-1, it can't do its job, right? It's like a key. IL-1 is like a key. If you weld a chunk of metal to that key, it can't get in the lock and it can't work, okay? So if you bind something to IL-1, IL-1 will not work as an inflammatory signaling molecule, right? So we could take an antibody and it would block the IL-1 response that I'm producing, right? So if I'm producing an inflammatory IL-1 response, we could inject that mouse antibody to human IL-1. It will bind to the human IL-1 and it will block its response. Isn't that amazing technology, right? So that's called a biologic drug. A lot of them are antibodies, but not all of them. And then a small molecule drug, again, it's just a few, like it's a few atoms here, right? Like this would be measured in the thousands of Daltons, so kilodaltons. This would be measured in the hundreds of Daltons. So they are orders of magnitude different on scale and on size. So here's an example here. We've got an ion channel here. So this might be, for example, a calcium ion channel. So it opens up and it lets calcium flow through. We can block that with a drug. So here's the small molecule drug here. You can kind of see it through there, but this is it zoomed in. And essentially the small molecule has gone into the hole. So this protein here is a protein that sits in our membrane and lets calcium through a small hole in it. It's an ion channel. This drug has essentially gone into that hole and plugged it like a cork. So now calcium cannot get in and through. So it's blocked the receptor and it's now interfering with that biological process. So this might be called a calcium channel blocker, which we use in the clinic regularly for various things. Heart problems and such. So this is how a small molecule works. It finds a little pocket. It's like a key, a lock and key. It finds a little pocket, it interacts with it, and it either changes the shape of the protein or it blocks the process from happening. Now, biologics are massive. They do something a bit different. So in the pale blue here, we have the interleukin-1 receptor, right? So interleukin-1 is an inflammatory cytokine. It binds to receptors, right? And activates those receptors to induce the inflammatory response. In the light gray here, we have interleukin-1. And in the red and brown over here, and in the yellow over here, we have two biologic drugs. These are antibodies that have been raised to bind to the IL-1 and prevent the IL-1 from binding to the receptor. You can see this is flipping massive. And if you imagine receptors are like a lock, and a ligand that activates that receptor is like a key that goes into the lock. If you change that key a little bit, you'll stop its function. Never mind attacking on a giant hunk of metal so it can't even get into the lock. That's how these biologic drugs work. Canakinumab. Canakinumab has just had an amazing clinical trial with amazing results that I wish I could go into, but we don't have time. So let's compare those. So small molecules, what are some of their advantages? Well, they're cheaper to make, right? It's just a simple chemical reaction and you can create bucket loads of this small molecule drug. Aspirin, you know, it costs pennies to make aspirin, right? It's cheap as chips. It's easy to distribute. So often because small molecules are really stable, they're robust, tiny molecules that can't really be attacked by anything, we can just pack them up into a dry pill and ship them all over the world, right? So they're very easy to distribute globally. They can be designed to cross body barriers. What does that mean? Well, you can design a small molecule drug to sneak across your stomach, to sneak across your intestine into your bloodstream. It could then cross the bloodstream into your brain. It could get throughout your body. You can design the drug in such a way that it can permeate throughout the body, so it can cross body barriers. It's easier to administer. It's easy to administer. You can just pop a pill to take a lot of small molecule drugs. You could also inject it. Suppository, there's loads of ways to take a small molecule drug. So it's cheaper to make, easy to distribute, it crosses body barriers, and it's easy to administer. What are some of the cons? Is that they can often be non-specific, and that's because you're trying to develop this tiny molecule to do this one thing, but it may look like other biological molecules. It may be able to fit into other locks, right? So the key that is the small molecule might be able to fit into other locks and cause problems. It may not be possible. That's an interesting one, right? We could do all the drug development in the world, and it may be impossible to target one receptor over another. Perhaps those locks, right, are so similar that it's very difficult to develop a key that will only unlock one of those locks and not unlock the other. There's no universal principle that says you should be able to develop a small molecule to target that protein specifically. And because of this, it also has lengthy drug development. So because there are so many ways a small molecule can interact with the body, it often requires, rounds and rounds and rounds of drug development. So you do the cell culture plate, then you jump into in silico, then you go back to in vitro, then you jump into in silico, then you go to an animal model and you find out it's toxic. So then you jump back into in silica, then you jump back into cell culture, and around you go. It might take forever. And in fact, you may never get there, and it may not be possible, right? So these are some of the disadvantages of small molecule drugs. Biologics, on the other hand, have small drug development times often, and they're very specific. Why is that? Because many of them are antibodies. Antibodies, by their very nature, can be developed to be incredibly specific, and because they're produced by a biological process that has evolved over millions of years to be specific and bind with high affinity and, just work, because we're really taking advantage of an already existed optimized system that is antibody generation, it can actually be a very small time, short drug development times, and it can be very specific because that's the way antibodies are. Now, some of the disadvantages is antibodies struggle to cross bodily barriers. They're a big protein. So how do they get inside a cell, for example? How do they get into the brain? How do they get it across the intestine? They're expensive to make. Antibodies are ridiculously expensive to make often. They require a purification process, often cell culturing. You can't just make them through a mass chemical reaction. They're difficult to administer. That's because you can't take them orally. Your stomach, your intestines are full of proteases that are designed to break down proteins. So you can't take antibodies orally. Typically, you need to be injected them. And they can induce immune reactions. Why is that? Well, because they're a biological molecule, your body might recognize them as a foreign antigen and have an immune response to the drug itself, okay? So it can induce an immune reaction, unlike a small molecule, which your body is unlikely to have ever exposed to, and it's unlikely to look like anything else that it's been exposed to, right? So, this small molecule here is unlikely to look like a pathogen, whereas an antibody is much more likely. So let's look at drug development in antihistamines. So remember, this is designed to block the histamine receptor, to block those sort of vascular and immune responses to histamine, to prevent these kinds of allergic reactions from happening. So histamine is a G protein coupled receptor. It's a receptor on the cell's surface membrane. The histamine receptor, and histamine will bind to it, cause secondary signaling throughout the cell, cause genetic changes, cell proliferation, vascular changes, that kind of thing. So it causes immune and vascular system inflammation. That's what we know it for, right? Histamine causes that inflammatory response. But not a lot of people know histamine is also a neurotransmitter that signals alertness, right? So, an interesting thing about histamine in the body is it's not great at crossing the blood-brain barrier. So if your mast cell releases a bunch of histamine, it's not great at crossing the blood-brain barrier and getting into your brain. Let's have a look at this. So this is a fluorescent chemical that was injected into the vascular system, and in red we've got neurons, and they've got the nervous system going on. And what you can see is that there's a difference there. The green has not leaked out into the brain, and that's because of this thing called the blood-brain barrier, where basically chemicals in the blood aren't allowed to freely cross into your brain. That would cause a huge bunch of problems, right? So we have this thing called a blood-brain barrier, which basically means the brain gets to control what crosses the blood-brain barrier. So it's very interesting. This is a fascinating thing, right? Histamine, likely developed as a signaling molecule, either in the brain or in the immune system first, right? So let's say it developed in the immune system first. Histamine now causes a massive immune response, and it causes inflammation, and it causes all those things, right? So now we have histamine and a receptor acting as a signal and a receiver of that signal. Now, evolution is incredibly lazy, which is very, very interesting. And it's because creating a gene from scratch is incredibly difficult and unlikely, right? Evolution essentially relies on chance events being beneficial. And so creating a gene out of whole cloth is incredibly unlikely. So what does evolution like to do? It likes to beg, borrow, steal. It likes to copy and edit. So what's happened here is one system has copied the gene into another system in the signaling molecule, but because they're separate, so the histamine signaling in your brain is separate from the histamine signaling in your immune cells, one has been stolen from the other, but because they're separate, they can now do completely different things. So histamine causes alertness in the brain, whereas histamine causes inflammation in the periphery. It's an amazing thing, and we see this all the time in evolution. It likes to copy and edit, it's like a bad student copying his neighbor rather than create stuff from whole cloth. So the first generation of antihistamines go into the histamine receptor, and like a key that is incorrect for the receptor, it goes into the lock, but it can't open it, right? So this is what an antagonist is. An antagonist goes into the receptor, but doesn't activate the receptor. But it occupies the receptor, so an agonist can't come in and turn it on, right? So histamine can't come in and activate the histamine receptor when the antihistamine drug, small molecule drug, is now sitting in the pocket of the histamine receptor. So it acts as an antagonist, like an incorrect key in the lock, the door won't open, so the correct key cannot get into that lock and open the door either. Now, the first generation of antihistamines could cross the blood-brain barrier. What that meant was this drug crossed the blood-brain barrier, blocked the histamine signaling in the brain. Now remember, histamine signaling in the brain causes alertness, so antihistamines used to be very drowsy and they cause sleepiness. So then through cell culture and in silico development, they developed the second generation of antihistamines, and they designed these to not cross the blood-brain barrier. So now you could take these antihistamines, they would be non-drowsy because they could not get in the brain to block the histamine signaling, but they would still block the immune response, which is flippin' fascinating and a stroke of genius. However, phase 4 clinical trials revealed something. People were dropping dead at a very low rate, an incredibly low rate. People were dropping dead on these second generation antihistamines. Terfenadine here is one of those examples. There was 2 actually that were produced in the 90s. And so people were taking these non-drowsy antihistamines and they were dropping dead. What was happening? Well, it seemed like there was an interaction with grapefruit and erythromycin. These 2 seemed to block the processing in the liver. So we ended up with really high concentrations of the drug in our blood. And what we found was at a certain concentration of that drug in our blood, it can cause your heart to sort of give out. So here is a normal heartbeat, boom, 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 boom. That looks fantastic. Here is it on terfenadine, boom, 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 ventricle fibrillation, right? So an absolute failure, a spasm of the heart. And it turned out this drug was interacting with a particular ion channel, which massively disrupted heart function. And now every drug that gets developed has to get tested against a range of ion channels, specifically in your heart, at a very high concentration to ensure that this doesn't happen again. But a critical thing here is that the phase 4 clinical trial system worked. This was such a rare event, it could not have been picked up in regular clinical trials. And it would be unreasonable to expect a clinical trial that could detect this when it was the first time that it had ever happened. But phase 4 clinical trials, government monitoring throughout the world, noticed it was happening, did something about it, and stopped it. It's a sign the system is working. And another example of this is the AstraZeneca COVID-19 vaccine. Now, the AstraZeneca COVID-19 vaccine was tested on was tested in a clinical trial of 33,000 people approximately, making it one of the most vigorously tested vaccines in history. That's an important point. A lot of people think these vaccines were rushed. They were not rushed. What happened is normally the journey has a lot of difficulties. A, funding, right? B, recruitment. Who enters clinical trials, right? Are you going to go out and enter a flu clinical trial? It's very hard to recruit that number of people. So what we found was when this pandemic hit, administrative barriers disappeared, funding problems disappeared, and recruitment problems disappeared. We were able to recruit thousands of people to a stage 3 clinical trial incredibly quickly. We also ran the stage 2 and stage 3 clinical trials side by side because we didn't run one after the other. We ran them side by side because we needed these vaccine spares as soon as possible. But a critical thing is these vaccines were essentially tested more rigorously than any vaccine in history because the clinical trials were bigger than most vaccines ever in history for a phase 3 clinical trial. So there's 33,000 people in this clinical trial, and they found no blood clots were occurring. Now, in phase 4 clinical trial, once the vaccine was rolled out, we realized that a rare kind, actually it's almost an autoimmune disease of blood clots was occurring following these vaccines. And it resulted in around about 6 deaths, I believe, in Australia so far out of 8.8 million doses. So it's an incredibly rare event. Maybe around 1 in a million people die from this, although that might be less because 8.8 doses might correspond to around about 4.4 million people. So it might be a bit higher than 1 in a million, but not much more. But the key thing there is to detect a 1 in a million event, you would have to give it to millions upon millions of people, okay? So you would need to run a clinical trial of millions of people. Now, it costs billions of dollars to run a clinical trial of 33,000 people. So how much would it cost to run a clinical trial on millions upon millions of people? It would essentially halt all drug development, which is why we have this the stage 4 clinical trial system, where we approve it for use, it goes out for use, and we monitor it closely to change prescription habits depending on adverse events that occur. So that's a really important point. The fact that we now know AstraZeneca causes clots, and the fact that we can do something about it and weigh up the risks and benefits depending on the pandemic conditions, like if thousands of people are dropping dead from COVID-19, it's probably worth it to get vaccinated to avoid that risk when the death rates are around 2% compared to 1 in a million, right? So it allows us to make informed decisions with this phase 4 clinical trial. But the important thing is these clots aren't the detection of these clots isn't a failure of the medical system. It's a sign that it's working, right? We got through phase 3 clinical trials, we produced these vaccines, and now we spotted that one of those vaccines, J&J, was the same, so maybe 2 of those vaccines were causing clots, while Pfizer and Moderna vaccines are still producing amazing results. And so, moral of the story is go get vaccinated. I had my Pfizer vaccines, I had almost no side effects, and it was just, honestly, I felt like I was watching the moon landing. I just felt like this was peak humans. You know, we hit a pandemic, and in a record time, we developed amazingly effective vaccines using brand new technology. And honestly, I was just proud as punch to be a scientist the day I got vaccinated. Anyway, so that's drug development. If you have any questions, pop them in the comments below, and thank you for tuning in.